Hello, physiology students. Okay, this is going to be our lecture for Monday. It's going to be August, 24th, and we're starting chapter 2. We're doing part 1, which is cell metabolism or overall body metabolism. Okay, so what I'm going to do is I'm going to share my screen and I'm going to go over the PowerPoint with you and all the other resources that we have to offer. Okay, so sharing screen. Here is our PowerPoint. So what we're looking at today is types of cell metabolism. Now, there are molecules that our bodies pull from our digestive tract, things that we eat and consume, and then our body metabolizes these once they're in our bloodstream. So number one, our body metabolizes carbohydrates. That is how our physiology is organized. Our bodies, our cells using carbohydrates the best. Carbohydrates have been getting a bad rap, but we absolutely need carbohydrates. And so when we're talking about carbohydrate metabolism, we're talking about making or breaking down, let me get a big screen here, making or breaking down carbohydrates. That includes glucose, which is a monosaccharide, a simple sugar, and that is the energy currency of our body. Glucose is what's floating around in our bloodstream. And then the storage form of glucose, known as glycogen, which is stored in our liver and our skeletal muscles and other organs. Now, we learned about glycogen in chapter 1 when we learned that the liver stores glycogen and will release glycogen, break it down into glucose, and dump it into the bloodstream if the hormone signal of glucagon is present. So this is a little bit of review. Okay, so our bodies are designed to metabolize carbohydrates best and first. And after that, our bodies can metabolize lipids. Okay, so if you think about this, when our bodies burn through all of our glucose stores and then all of our glycogen stores, what our bodies are going to do next is turn to our fat stores. That is our storage form of energy when we need it. So lipid metabolism is the making or breaking down of lipids, and we'll talk about that in a little bit. So in addition to metabolizing these compounds, our body can metabolize protein. Okay, so protein metabolism includes the making or breaking down of proteins, amino acids. Now we make proteins all the time from simple amino acids in our cells, but we also can break them down if we need to. In fact, if you think about it, if our body runs out of carbohydrates, it'll turn to lipids next. And if we ran out of all of our fat stores, let's say we were in a starvation situation, next our body would turn towards our muscle and break down that muscle for energy. People who are basically anorexic or who are so sick that they're not eating or they're in starvation mode, you'll start to see muscle wasting. Their muscles will shrink down because their bodies are using that for energy. Okay, next, our bodies routinely metabolize lactic acid. So we're going to talk about that. But lactic acid is a byproduct of routine skeletal muscle activity and can also be the byproduct of cells metabolizing in the absence of oxygen. But I'm getting a little ahead of myself. So let's start at the very top with carbohydrate metabolism. Okay, well, glucose metabolism, we'll talk about that in detail. You probably learned that in intro biology, and that is going to be glycolysis, or the use of glucose for making ATP during cell respiration. Now, that's a long process, and we'll get to that shortly. But that's glucose metabolism. That's why our bodies need glucose. Okay. Glycogen metabolism, we'll go a little bit back to chapter 1 when we talked about glucose, blood glucose regulation and the negative feedback pathway. But glycogen metabolism includes making glycogen from glucose precursors to breaking down that glycogen when our bodies need it. I mean, that's why our bodies store glycogen. It's an energy resource if we happen to need it, if we're skipping some meals or something like that. So glycogen metabolism will include, oh, it's not showing up here, the making of glycogen. So look at the word glycogenesis. Oop, back up. Glyco is for, well, the word glycogen. Glycogen is in the word. If you back up a little bit and look at genesis, the word genesis means, I'm going to have to escape out of this. Genesis means to create. Okay? All right, so let's back up out of that. So if you look at it, glycogenesis, is the making of glycogen. Okay, so genesis means create. Now, that's making glycogen from glucose precursors. If you want to break glycogen down, it's called glycogenolysis. That's not how it's pronounced. It's glycogenolysis, but it's got the word glycogen in it, then an O, and then lysis. Now, lysis means to break apart. That's what lysis means. Okay, so the lysis of glycogen is the breaking apart of glycogen to release glucose. So these words have meaning. Genesis means create, lysis means to break apart. Okay, so question. What do you remember from chapter 1 about this process of glycogenolysis? What pancreatic hormone can stimulate glycogenolysis in the liver, or the breakdown of glycogen into glucose, which is released into the bloodstream? First of all, why would the liver need to break down glycogen into glucose and to release it into the bloodstream? Number one, it's when there's not enough blood glucose. Blood glucose is too low. So the hormone from the pancreas, think about this, glucose is gone. So the pancreatic hormone is glucagon. All right, glucose is gone. So when glucagon is in the bloodstream, the liver will engage in glycogenolysis, breaking apart its glycogen stores into glucose and releasing that glucose into the bloodstream to bring blood glucose back up to normal levels. Okay, so that is part of the negative feedback. Okay, backing up a little bit more. Okay, just go back to chapter 1. What pancreatic hormone would stimulate glycogenesis? Okay, making of glycogen from glucose. Well, first of all, the liver would have to take in glucose from the bloodstream in order to convert it into glycogen. So what pancreatic hormone tells body cells, including liver cells, to take in blood glucose and to use it in metabolism? What is that? If you said insulin, you're right. Insulin tells body cells to take in glucose and to use it, and that includes the liver taking in glucose, which it will then use for its own metabolic needs. But above and beyond what the liver needs, it will take that extra glucose and make it into glycogen and hold on to it. Okay, lipid metabolism. Okay, making or breaking down lipids. So if you remember, genesis means create and lysis means to break apart, it becomes very simple. So lipogenesis is the making of lipids, and you make lipids from extra glucose. Okay, so we'll learn that process. But when you have above and beyond the glucose that your cells need, your liver will take it, convert it into glycogen, and hold on to it. But your other cells, like fat cells and the liver itself, will take extra glucose. In addition to converting into glycogen, it'll convert it into fat. Right? That's when you have too many calories, you start to gain weight. Okay, so that's lipogenesis. You take extra glucose and you turn it into lipids, and those lipids include ketones, fatty acids, and triglycerides, and that's your storage form of energy. Now, if your body needs it, it can take those fat stores and break them down into things like, well, back to fatty acids and ketones and even glucose. And that's called lipolysis. So it's got lipo for fat, and lysis means to break apart. So you break down triglycerides into fatty acids, which you can use for energy, into ketones, which you can use to energy, and you can even take basically fats and turn them back into glucose, because that's where they came from in the first place. All right, so when you took chemistry and you looked at all these chemical reactions, you always looked at a reaction, this plus this equals this, right? You think of chemical reactions going in one way, but guess what? They can go backwards too. That product can be broken down in its component parts, and this is what metabolism is all about, is the reaction is going either way. Okay, so let's look at glycogen metabolism. Okay, either making glycogen or breaking it down. So glycogenesis, genesis means create, okay? So it's creation of glycogen from glucose. And what you do is you take glucose molecules and you use an enzyme called glycogen synthase that makes those glucose molecules into glycogen. Well, think about this. It ends in ASE. All right, so let's get out of this screen and I will write this down. If it ends in ASE, that stands for an enzyme. Oop, there we go, enzyme. And synthesis means to create as well. It's another word for creation. Okay, so if you look at this, the enzyme, ASE, that synthesizes, and the word that it's producing is in front of the enzyme, glycogen. So it literally means the enzyme that synthesizes glycogen. Okay? All right. Now, that's glycogenesis. Now, that happens if there's insulin in the bloodstream and the liver takes in glucose and uses it for its metabolic needs. And if it's got more glucose than it needs, it'll turn it into glycogen and hold on to it in case it needs it. Now, if the hormone glucagon is in the bloodstream, the liver will break down glycogen in a process known as glycogenolysis. Okay, so it's a two-part step for the liver. It's a one-part step for your skeletal muscles because your skeletal muscles store glycogen and your liver stores glycogen, but your liver will share the breakdown of glycogen, which is glucose. The liver will share glucose with the bloodstream, but the skeletal muscles are greedy. They can break down glycogen that they're storing, and they will only use it themselves. They won't spill that glucose into the bloodstream. So skeletal muscles will never share glucose with the body. But here's the process of breaking down glycogen when you need it. It's there in case you need it. So you start off with glycogen, and it becomes an intermediate glucose called glucose 6-phosphate. And that's what your skeletal muscles use. Most of our cells can use glucose 6-phosphate as its energy source. To break glycogen down, you need the enzyme called glycogen phosphorylase. It ends in ASE, and it's got the word phosphoryl. And if you phosphorylate something, you basically cleave a phosphate off it, or you kind of break it down. And it's telling you the word in front of the enzyme tells you what it's processing. So it's processing glycogen. So it's the enzyme that phosphorylates glycogen or breaks down glycogen into glucose 6-phosphate. Now, the liver and the skeletal muscles both have this enzyme, and this glucose 6-phosphate can be used inside of cells, but it can never leave a cell and go into the bloodstream. In order to do that, you have to break glucose 6-phosphate down one more step, and it requires an enzyme that only the liver has. So glucose 6-phosphate gets broken down by glucose 6-phosphatase. So ASE is the enzyme, and the word in front of it is telling you what the enzyme is breaking down. It's glucose 6-phosphate. It's like the whole word, glucose 6-phosphate, but they cleaved off the E and call it glucose 6-phosphatase. So that's telling you this enzyme is breaking down glucose 6-phosphate into free glucose. Here's another way to remember this, okay? If you remember this two-step process, there's two enzymes. Look at the first enzyme, phosphorylase. It's got an L in front of the A's. And then over here, phosphatase, it's got a T in front of the A's. What comes first in the alphabet, L or T? So phosphorylase comes before phosphatase. And if this was on a multiple choice exam, if you just remembered L comes before T, the phosphorylase, the enzyme early in the process, and the phosphatase is later in the process. Okay, so let's move on. Okay, so I provided on your syllabus, the online syllabus, a blank flow diagram for glucose and glycogen metabolism and the key. So knowing that glucose is turned into glycogen is a process called glycogenesis. Do you remember the enzyme we just said that synthesizes glycogen? It'll end in ASE. It has the word synthesis in it, and in front of the enzyme, it has the name of the molecule that's being processed. It's glycogen synthase. Okay, down here for breaking down glycogen or glycogenolysis, the first breakdown involves the letter L in front of the A's, phosphorylase, and it's going to have glycogen in front of it because glycogen's being processed. It's glycogen phosphorylase. And then over here, this is where the T comes before the A's, and it's got the word glucose 6-phosphate in it. It's glucose 6-phosphatase turns this intermediate glucose into free glucose. So I recommend practicing this flow diagram because I'm definitely going to ask you on a future exam. You need to know these enzymes. Okay, let's move on. Okay, here we come. Glycolysis. We all had an intro bio. We all hated it because you had to memorize all these cycles, all these acids. So I've tried to simplify it. And so pages 27 and 29 of your wiki text. So let's go to that right now. 27 and 29. So let's type it in. Let's type in 27. I want you to get used to doing this. Oh, look at that. It's right there. Let's move this out of the way. Okay, cellular respiration. And what's my dialog box? Read this section and the rest of the chapter as part of chapter 2, part 1. You will learn more about cell respiration and ATP production. So it talks all about glycolysis, the Krebs cycle, lots of steps. You're not going to have to know all that detail. And then the electron transport chain. And I'm not even going to talk about the redox reaction. But if you want to read more about carbohydrates and lipids and proteins, that follows. But I'm not going to go into that detail. But that is there for you if you want. That page is there because you can read the corresponding material in the wiki textbook. So let's start with glycolysis, and this we'll get through for Monday's lecture. So this is the use of glucose to make ATP. That's what glycolysis is. Okay, it's the first step in cellular respiration. So it occurs in your cell's cytoplasm, and cell cytoplasm is the fluid portion of the cell that surrounds all the organelles and everything. And really what happens is I made a simplified diagram right here. One glucose molecule will go through glycolysis, and what you'll get is the main product comes out at the bottom, 2 pyruvate molecules, or pyruvic acid. I call it pyruvate. And side products of 2 NADHs, so these are 2 hydrogen ions, actually 4, and a little bit of 2 ATP. That's a little bit of energy. Okay, so let's fill this in right here. So what you've got is the main product is 2 pyruvate, okay? And I will make that bold because it's the main product. Side products include 2 NADH2 and then a little bit of ATP, 2 ATP. And for those of you who are really more attentive about this stuff, I know that in glycolysis, you actually make 4 ATP, but 2 ATP are used up in the process, so you only get 2 net ATP. So if you learned that, yes, I know that, but what you have to use is 2 ATP as a result. And now that pyruvate can go one of 2 directions. If there is oxygen, that pyruvate can go through 3 more steps: pyruvate conversion, Krebs cycle, and the electron transport chain, and you can make a lot of ATP. If there is no oxygen, your cells can process pyruvate, but it'll only make 2 ATP and some lactic acid as a byproduct. So oxygen is always best. So let's follow what happens after pyruvate is made. So this is just to give you some perspective here, okay? Glycolysis produces pyruvate. Pyruvate can go one of 2 directions. If oxygen is present, it'll go this way. If there's no oxygen present, it'll go this way. Now, if there's no oxygen present, your cells will respire in the absence of oxygen. That's called anaerobic respiration. Most of our cells can do this, but for most cells, especially heart cells, brain cells, and other tissues, if they're forced to go through anaerobic respiration, it's going to be an emergency situation. Now, our skeletal muscles can do this routinely. If you've ever exercised and pushed yourself past a certain point, you'll start to feel that muscle burn, right? And eventually, if you ignore that and you keep pushing your muscles, they will completely fatigue and give out, right? That burn is showing you've pushed your muscles to the point where they've run out of oxygen, they've probably run out of glucose, and now what they're doing is they're respiring anaerobically, and they're taking pyruvate and they're fermenting it, and they'll produce a just poor amount of ATP, not enough to really keep you going. That's why at the gym you have a spotter when you're really pushing your muscles, because you're pushing them past the point where you've used up all your oxygen, and those muscles are building up burn, which is from lactic acid, that's producing the burn, and you're not producing enough ATP to keep that muscle activity going, and eventually those muscles, they say, that's it, I'm done, okay? So even skeletal muscles run out. They can ferment and produce a little bit of lactic acid and ATP, but they're gonna quit. Now, if you have a loss of oxygen in the blood supply to your heart or your brain, that's an emergency. Those organs use so much oxygen and glucose that they cannot go without either of those two very long without cell death. So, in your heart, in your brain, if you lose blood supply to your heart is constantly beating, and it needs constant supply of oxygen and glucose. If you have a lack of blood supply, which is called ischemia, to the heart, your heart will eventually, that part of the heart that's losing blood supply and oxygen, it will resort to anaerobic respiration. It'll produce a little bit of ATP and lactic acid, and that ATP is not enough to keep the heart going, and those heart muscle cells will die, and the lactic acid produces a burn that a lot of men who are having a heart attack will feel that burn going down their left side of their chest and down their left arm. Women don't seem to have those quite the same symptoms, but that pain is from lactic acid production from heart muscle that's not getting enough oxygen, not getting enough blood supply. In your brain, if you have an interruption of blood supply to the brain, which is known as a stroke, the same thing will happen. Your brain cells will resort to anaerobic respiration. They'll produce some lactic acid, which could be measured in the bloodstream, and just a little bit of ATP, but not enough to keep your brain cells alive. Brain tissue will die in a stroke. So our cells don't like being forced to anaerobically respire. That's not the way they're designed. The way they're designed is pyruvate, when oxygen is present, goes through pyruvate processing, the Krebs cycle, and then the electron transport chain, and as a result of one glucose molecule, you'll make as much as 34 and possibly up to as much as 36 ATP. Now we're talking. That's the kind of ATP that keeps our cells happy, okay? All right, so let's go on to the next slide. So this whole idea of a loss of blood supply, which is called ischemia, any organ or tissue can have a loss of blood supply or have an ischemic event. But this is relevant to nursing. So this whole cell metabolism thing, this whole glycolysis thing, is it relevant to everyday nursing? It sure is, because if certain tissues are deprived of oxygen or deprived of a blood supply through an ischemic event, they're going to resort to anaerobic respiration. They will produce lactic acid as a byproduct and not enough ATP to stay alive. So there's a clinical act, okay? But let's define ischemia here first. Ischemia is a loss of blood supply. That's a general term, okay? Loss of blood supply. Now let's move this. Okay. A little bit more. There we go. Okay, so let's look at the clinical app. It'll take a second for it to pull up. But when you're looking at the PDF of this lecture online, you'll be able to pull up this clinical app and read it. But here we go. Ischemia refers to inadequate blood flow to an organ, such as the rate of oxygen delivery is not enough to maintain aerobic respiration. Most of our metabolically active tissues need so much oxygen. If you deprive them of it, sure, they can resort to anaerobic respiration, but they can't keep that up without dying. So when your heart muscle doesn't have enough blood flow, it's called myocardial. Myo means muscle, cardio refers to cardiac. Ischemia, literally, interruption of blood flow to the heart muscle, okay? If there's a blockage in a coronary artery or something like that. So if the loss of blood flow is prolonged, part of the heart will die, and if it's bad enough, you can die from it, okay? So it can lead to the pain that you experience called angina pectoris, severe pain in the chest and sometimes radiating down the left arm, could be a prelude to a myocardial infarction or a heart attack. So those kinds of signs, those kinds of pains should never be ignored. Okay, so let's go back. So loss of blood flow to the heart. Without arterial blood and oxygen, the heart switches to anaerobic respiration, makes a little bit of ATP and some lactic acid, which can produce that burn that you feel with angina pectoris. Oops, let's back out. Okay. Pain associated with loss of O2 supply to the heart. It can be a prelude to a heart attack, a myocardial infarction. Now, the reason I bring this up is there are lots of markers that you can look for. So someone comes in, they're suspected of having a heart attack, they complain of pain, which is angina pectoris, and you suspect they have a heart attack. So you're going to do all the tests. You're going to hook them up to an EKG. You're going to see what's going on with the heart. You're also going to draw blood and send it off immediately for analysis. You might even put some through a CT scan and see if they've got a blockage in the blood flow to the heart. But there's lots of things you can look at, and we'll talk about creatine phosphokinase or CPK values. But what they're finding out is that when a tissue is deprived of oxygen, the blood levels of lactic acid or lactate will rise very quickly. And you can actually use serum or blood lactate levels as a marker for if someone's having an ischemic event. And that levels of lactate will rise faster than the other factors in the blood that can rise with a heart attack. So normal serum lactate, know it as nurses, okay, between 1 to 0.5 millimoles per liter. If lactic acid or lactate in the blood is higher than that, it's called hyperlactatemia. Emia means in the blood, it's got the word lactate in it, and hyper means excess. Hyperlactatemia means some tissues in the body are going without oxygen. Now, I'm not going to pull it up for you, but if you want to look at a nursing journal article, I've got a reference here where you can talk about how you can use serum lactate as a marker, an indicator of an acute heart attack. Okay, so this whole glycolysis and how our cells metabolize glucose and whether it goes through the anaerobic pathway or the anaerobic pathway is incredibly relevant to nursing and human health. Okay, let's move on. Okay, aerobic respiration. So after glycolysis, so glycolysis, remember, is glucose is broken down into 2 pyruvate molecules, and then you have side product molecules, 2 NADH2s and 2 ATP. But let's focus on that pyruvate. Pyruvate can go one of 2 directions. If there's oxygen, it'll go through these 3 steps I'm going to talk about. If there's no oxygen, then it will go through anaerobic respiration, make 2 ATP, and make lactic acid. And so we talked about that. So let's talk about after pyruvate is produced and there's oxygen present. The first thing is that pyruvate is going to be processed. It's called pyruvate processing. So it's telling you it's processing pyruvate. It's also known as pyruvate conversion. I don't know what you learned it as, but it's known as pyruvate processing. So what happens is 2 pyruvate enter, and what I put in red here is the main product that can go on to the next step. So the main product's in bright red. Now the side products are, look, oh, some more, 2 NADH2s. That was produced from glycolysis too, okay? We'll talk about all those hydrogen ions shortly. And then you get another byproduct called CO2. CO2 is a byproduct or waste product of cellular metabolism. That's why we breathe out CO2 to get rid of it. But look at the bright red molecule. 2-acetyl-CoA is going to go on to the next step, the Krebs cycle. That's known as the citric acid cycle. That's the cycle where you had to learn all those acids. Well, I'm not going to expect you to learn all those acids. I don't even remember all those acids. If I needed to know them, I would open a textbook like a normal human being. But what I do know is that 2-acetyl-CoA here enter the Krebs cycle. And each acetyl-CoA will spin the cycle twice. And what comes out at the end of 2-acetyl-CoA entering is 2 ATP. You get a little bit of energy. You get 6 NADH2s, so that's a lot of those. And you get 2 FADH2s. Notice I put the hydrogen ions in red. The hydrogen ions produced from glycolysis, from pyruvate processing, and from the Krebs cycle. All those hydrogen ions are going to go on to the next step. So chemistry. You had to take chemistry to be in here. Okay, chemistry. Hydrogen ions. They have 1 proton, no neutrons, and 1 electron. So hydrogen ions have electrons. Those electrons are going to be used in the next step, which is the electron transport chain. Okay, so all those NADHs, okay, I'm actually missing 2. There's an additional 2 that I should have here. In fact, let's fix this. We'll do 2 NADH2. Okay, we'll do comma. Oop. lost it. Okay, and a comma. Okay, here's what's going on. And I'll make it big here again. So the two NADH2s that were produced from glycolysis are there, those H's. The the two NADH2 from pyruvate processing, the two NADH2 from glycolysis, and the 6 NADH2 from Krebs cycle, all those hydrogen ions get shuttled along the mitochondria of your cells. And what happens is a lot of ATP is made. 30 to 32 ATP are made from the electron transport chain itself, okay? And then what we also have at the end as side products are 10 NADs, all right? 2 NAD plus 6 NAD is 8, plus another 2 is 10. And notice the plus sign means the hydrogen ions have been cleaved off of that molecule. Same thing with the FAD, it's 2 FAD plus. The hydrogen ions aren't attached to that anymore. Where did the hydrogen ions go? They bound to the oxygen that you breathe in. So what happens is you have 12 water molecules that are called metabolic water that are byproducts of cellular metabolism, basically aerobic respiration. So you get a whole bunch of ATP, some cleaved NAD and FAD, and you get metabolic water. All right, that is aerobic respiration. That's going to keep your cells alive and happy. Okay. So here is my own diagram, which is much, much simpler than the one you'll find in a textbook. I'm going to move this up here. So we have at the start, glucose goes through glycolysis. The main product is down here in bright blue, 2-pyruvate. Side products, 2-NADH2, 2-ATP. Great. Well, we'll come back to those NADHs later. Pyruvate, if there's no oxygen, goes through fermentation or anaerobic respiration to make a little bit of ATP, not enough to keep your cells happy, and lactic acid as a byproduct, and that usually is a sign that bad things are coming. Now, if oxygen is present, pyruvate enters pyruvate processing. It makes 2-acetyl-CoA as a main product in bright blue. Off to the side are side products, more NADHs, and some CO2. Now, the acetyl-CoA enters the Krebs cycle, and what you get from the Krebs cycle, primarily as a main product, are hydrogen ions, 6 NADH2, 2 FADH2, that's the first time that FADH2 makes an appearance, more CO2 waste, and a little bit of ATP. Okay, so notice in this flow diagram, it's a lot of 2 of everything. 2 ATP, 2 pyruvate, 2 acetyl-CoA, 2 NADH2s, right? The only time that you see 6 NADH is with the Krebs cycle. And here's what I remember with the N. The N, it's not what it really stands for, but I think N means more numerous. There's more NADHs in this whole process. They're more numerous. The FADH2 only makes an appearance in the Krebs cycle. Think fewer FADHs. You'll only see 2 of those. Okay. Hydrogen ions from up here, here, and here enter the electron transport chain. The main product is going to be, move this out of the way. Here we go. How about over here? It's going to be a lot of ATP, tons of ATP, 30 to 32 ATP. The cleaved NADAs and FAD, and then the metabolic water. Okay, so aerobic respiration total of 34 to 36 ATP. Where did I get that? I added 30 plus 2 plus 2 is 34. Now, sometimes you can actually produce a little extra ATP and it gives you 36. So you probably learned it as 34 ATP. Count up the ATPs in the aerobic respiration pathway. That's a big bang for your buck. 1 glucose molecule and you get 34 ATP. Wow, that's efficient, okay? Okay, so that was glycolysis from the start of 1 glucose molecule to a bunch of ATP. All right. Now, here is a GIF of the electron transport chain, and what you've got are hydrogen ions, okay? And they're shuttling across the mitochondria of your cell. Hydrogen ions are coming in. Look at that, okay? And I'll let this run again. Oh, there's your mitochondria, okay? Okay, so hopefully this will run one more time. We'll give it a second here. Deep inside your mitochondria, hydrogen ions enter the electron transport chain, okay? And what happens is Oxygen binds with those hydrogen ions and makes metabolic water, but what you make is a bunch of ATP, a lot of ATP, okay? 30 to 32 ATP molecules. All right. So, someone asked me this question in grad school, and I didn't know how to answer, and I was embarrassed. And I bet if you ask some of your future nursing faculty, they might not know it. They might. Why do we need to breathe oxygen? And I thought, well, we need oxygen to breathe, to live. Our cells need oxygen. But that's not really it. Our cells need ATP. And what oxygen does is it acts as a final electron acceptor, hydrogen ion electron, in the electron transport chain and production of ATP. So those hydrogen ions that are being shuttled across your mitochondrial membrane, eventually as the ATP is spit out, right, those hydrogen ions will bond to oxygen and you get metabolic water. So oxygen acts as the final electron acceptor in the electron transport chain. So this is a big segment here, okay? And we're going to stop here for today, but I want you to look at the blank flow diagram, and I'll pull it up here. It'll take a second, and I want you to practice filling this out so that you can learn it, all right? I'll ask you what enters glycolysis and what comes out as a main product. What enters pyruvate conversion and what comes out as a main product? What enters the Krebs cycle and what comes out as the main product? What enters the electron transport chain and how much ATP is made and what are all the byproducts, okay? I can also ask you what happens to pyruvate if there's no oxygen. So you'll want to practice that flow diagram, okay? Let's go back to our PowerPoint. All right, so the last concept, cyanide. Most of you are not going to see people come into a hospital with cyanide poisoning because they'll probably die before they get there. Cyanide is a very fast-acting poison. You've probably seen movies where they have a spy, Russian agent, and he got caught by the CIA, and they're going to interrogate him, but he's got a cyanide capsule, and he eats it before he can be interrogated so he can't spill his secrets. Cyanide is very deadly poison, and if you click on the clinical app, it pulls up the cyanide clinical app reading about why it is so deadly. And so we'll wait for it to come up here. It's a fast-acting lethal poison that produces symptoms as rapid heart rate, lethargy or fatigue, seizures, and a headache, and that results in eventually a coma and death. So what cyanide does, it prevents oxygen from acting as the final electron acceptor in the electron transport chain. Now think about this. If you don't have oxygen acting as the final electron acceptor in the electron transport chain, you don't get to make, your cells don't get to make 30 to 32 ATP. It blocks that. So if your cells can't make enough ATP, it's just as if you're going through anaerobic respiration. You only get a little bit of ATP, but not enough for any of your cells to survive. They're all going to die, all of them. Your brain will go first, followed by your heart and everything after that. So cyanide keeps oxygen from basically accepting electrons to make ATP. So very deadly. Okay, folks, so that, I guess one more thing. Okay, so where are all these things happening? I promise we're going to stop at this point. So what we've got here is our liver, which stores glycogen, okay? And then we have our intestines and our stomach. So if you're consuming carbohydrates, right, glucose, glycogen, whatever, you can get glucose from your food, okay? And then that glucose enters the, from the digestive tract, crosses the lumen of the intestine, enters the bloodstream, which is what this is, glucose in the bloodstream. Or let's say glucagon from the pancreas was in your bloodstream and the liver breaks down glycogen in glycogenolysis. Remember the enzymes that are necessary for that process to happen? Go back in your notes and study it. But glucose is released into the bloodstream. Now we've got glucose in the bloodstream, and we've got a cell here. Here's the cell membrane. Now, what pancreatic enzyme is necessary to tell that cell to take in the glucose from the bloodstream? What was that pancreatic hormone? If you said insulin, you're right. So if insulin is present, the cell will take in glucose into its cytoplasm. So this light yellow is cytoplasm. That is where glycolysis happens. Glycolysis goes through, well, glucose goes through glycolysis and produces 2-pyruvate. Now the pyruvate can go one of two directions. If there's no oxygen present in your cell's cytoplasm, anaerobic metabolism occurs and you get 2 ATP and some lactic acid. So this can happen routinely in skeletal muscle, but if this was happening in heart or muscle cells, you'd be on your way to death. But if there's oxygen present, it's not shown here, but pyruvate goes through conversion, and when it goes through conversion, it produces 2-acetyl-CoA that enter the Krebs cycle, that make a bunch of hydrogen ions, that go through the electron transport chain, that make a bunch of ATP. So that's where all these things are happening, okay? All right, so this is a good place to stop for Monday. That was a whole bunch of information, and we'll pick up the rest of this information on Wednesday. All right, so I know I threw a lot at you, so definitely go through and review these notes. And if you have any questions, email me. We'll have a Zoom office hours this coming Monday at noon, so hopefully I'll see some of you then. All right, bye.